So welcome back to the afternoon session, and thanks to be here again. And it's to me an honor and a pleasure, I would say, you have to say, to introduce the next speaker. So Francisco Bezanilla. The title of his talk is.
You're ready to talk whenever you wish. You can.
Okay, whenever you wish. The title of the talk is Volta, the Sensors and Channel Opening. So please, Francisco. Francisco Bezanilla. The title of his talk is.
You're ready to talk whenever you wish. You can.
Francisco, please close Qvbr.
Opening. So. Is
you can.
Francisco, please close Qvbr.
Okay, I did.
Opening.
All right.
So,
all right. Thank you so much for the invitation. Can you hear me properly? Okay.
You can.
Francisco, please close Qvbr.
Okay, I did.
Opening.
All right. All right. Thank you so much for the invitation. Can you hear me properly? Okay, let me close Qvbr then.
Francisco, please.
Okay. I think now we're okay. All right. So I hope you're hearing me now. Thank you so much for the invitations, and I would like to thank the organizers. And what I would like to do is to talk today about voltage sensor and channel opening. But first, I would like to thank the people in my laboratory that have been contributing to this work. Especially, I would like to mention Gerald Lacroix, Joao de Souza, and Michael Priest, Elizabeth Lee, and Carlos Baceto. I'm going to put the pointer here. I hope you can hear it. Okay, laser pointer there. So just as a motivation, all of you know about this, of course, but this is actually what motivates us to do this work, which is the generation of the nerve impulse. And here we're showing the classical action potential of a squid giant axon and how the sodium conductance precede the opening of the potassium conductance, which repolarize the membrane. This is all due to the, of course, the fantastic work of Cochin and Huxley in 1952. Now, what do we have to make as a summary of the most important points about this work is that the conductance pathway have selectivity. You have a sodium pathway and a potassium pathway, meaning that they are selected to sodium and selected to potassium. And then the conductance has a very steep voltage dependence, and this is something that is going to be the main subject of this work today. So this is represented here in this cartoon, like the ion channel is opening only after these gates are moved by the voltage, which is the actual driving force for moving the gates. And the third important point is that the kinetic of the sodium conductance is way faster than the potassium conductance, which is really critical, so that otherwise we will have an action potential. You have to first let the sodium come in before the potassium goes out. Now, how about the gating current? How about this, coming back to this diagram here, how this voltage dependent originates? Well, that originates because there's a voltage dependence on the capacitance. So just coming back to the standard thing that everybody knows, that Q equals CV. When you charge or discharge the capacitor, you have to take the first derivative of that to give you the current. But that has two parts. The first part is CdVdt, which is what we normally use in our capacity current. Everybody knows that. There's a linear dependence of the charge with a V, because you have plenty of ions that can go close to the membrane to separate charge from one side to the other. But there's a second term in this equation, when you take derivative of a product, of course, is BdCdt, and that one contains movement of charges enclosed in the proteins, which are the ones that we're interested today. And that Q saturated with B. Once they all move in the extreme position, then that's the end of the saturation, right? So that produces the gating current. So that's the movement of the voltage sensor, the gating current. I'm not going to talk too much about gating current today, but I'm going to talk about the origin of it. So how does the voltage sensor domain know it works? Today we know a lot about it because we have the structures and we have a lot of physiology done on it. So essentially, in a voltage sensor, we have 4 charges in the S4 segment. There are more of those, but those are the most important ones, for example, for shaky potassium channel, that are normally sitting in the intracellular space when in the intracellular cavity of the protein, when this is in the closed state, which is the negative potential. When one depolarizes the cell, then these charges move to the outside by depolarization, crossing this green region here, which is the hydrophobic plug. Okay, so resting to active require repolarization, moving the charge, and that produces, of course, the gating current. So there are 2 questions I'm going to address today. One is, what is the role of this hydrophobic plug in transferring these charges? And the second part is, what are the paths of these gating charges, right? So going to the structure, this is the structure of Long et al., the McKinnon structure, where you have the Kv 1.2. I'm looking from the top, from the outside of the channel into the region around the S4, S4, S3, and S2, and S1. And you can see here the first 3 charges, arginines, which are essentially outside, and they are completely blocked. They don't have any space to go through. So in fact, somehow these charges have to make room to go through this hydrophobic plug. There are 10 residues here that have been identifying this hydrophobic plug, which is narrow. It's only about 10 angstrom in size. So essentially, the charge moves across 10 angstrom of hydrophobic material, okay? So what we did was actually to go and explore all those hydrophobic residues, all this residue of the hydrophobic plug, by changing them to all the amino acids. I'm going to just summarize what we found. So in the case of the closed state, when the resting state, we found that there is a very important residue, which is a small residue. It's a serine here, 240, which what it does is introduce what is called a steric gap, which is pictured here in this diagram, where you can see is that the charges here normally are sitting in a hydrophilic region. I'm pointing here to the S240, and you can see the lines of isopotential are real mostly around the hydrophobic plug. But to get the first charge to be influenced by this field, you have to introduce into this region. So the fact that you have a small residue here is what you need to get this and engage into the opening of the channel, okay? And the activation of the voltage sensor. So if we go now to the intermediate state, let's say when the voltage with the Qv is in the center, there is a very important residue here, is isoleucine I287 in the case of the shaky potassium channel. Now, this is the one that actually makes the main energy barrier for crossing the arginine from one side to the other. And in fact, what we have found is that it is critical the type of residue you put here. In the case of the potassium channel, which are slow, it's an isoleucine. In the case of the sodium channel, in the first 3 domains, which are the fast domain of the sodium channel, here is a threonine. So it's hydrophilic. So what it does effectively is to lower the energy barrier, as I'm showing here, to cross the arginine from one side to the other. And here's a diagram with the waters penetrating into those cavities and where the I287 is, which is right in the center. Finally, in the active state, there are three important residues. It's a phenylalanine 244 to 90, which are actually doing sort of a spaced van der Waals clamps, okay, to hold it in the open position. And a real hydrophobic plug stabilization is done by the isoleucine 237, okay? So now, how about the gating charge trajectories? Well, one would like to have a path of the individual charges at the arginines, in this case, to the other plug. It would be nice to have a fluorescent arginine, right? Well, that doesn't exist, but we have something that we found that is really quite close, and it's this fluorophore called monobromo trimethyl ammonia bimane, Qvbr as we call it, is positively charged, has a bromine group here that can be attached to a cysteine. Second, it's comparable in length, you can see on the bottom here. This is the size of the structure of the Qvbr compared to an arginine. And it's fluorescent, of course. It's not environmentally sensitive, but it is quenched drastically by two amino acids, tyrosine or tryptophan, okay? So here's the idea. Suppose that we put now Qvbr instead of the charge. We replace it, and then we put tryptophans in the path. So if we put a tryptophan close to the beginning of the path, when you activate, the Qvbr will move away from it and produce an unquenching of the Qvbr. Now, if it's exactly in the middle of the path, you will see nothing because it will just cancel the on to the off. Where you had single molecule, you will see a jump, of course, but that's not the case here. In the activation case, then if you have the tryptophan on the other side, when the Qvbr plays close to the other side in the active state, you will see a quenching of the solution. So you have unquenching in one situation and quenching in the other situation. So in other words, by putting tryptophan in different places along the pathway, we could actually follow the path of the charges, in this case, the Qvbr. So here's a simple model for quenching. I mean, it's just essentially it's a proximity where lambda is about 4 to 5 angstrom. And so the fluorescence actually changes according to the proximity between the tryptophan and the Qvbr. So you have a many states in the transition between the active and between the passive and active. What you will see is a time course of fluorescence, according to this equation here. But this will have n-1 if n is the number of state eigenvalue. Therefore, now the actual time course of the fluorescence will be influenced by the position of the tryptophan, or the transfer of the charge in this case. So what the method is very simple. It's simple in principle. What we do is just mutate, for example, the first charge to a cysteine. Then we inject this RNA made from this DNA into the O site. Then we stain with Qvbr. And then we're going to set up where we can measure the fluorescence simultaneously with the voltage clamp measuring the gating current. Okay, so that we have a simultaneous recording of gating and fluorescence. So here's an example when we replace the first charge by making R1C into Qvbr, okay? Here's the gating current on the left, and here the QV curve, and here the signal. So they are actually an intrinsic signal that you can see as a quenching of the fluorescence. Now, if you look at the structure that we know, which is the open active state, relaxed state, you can see that the tryptophan in position 454 would get very close to the first charge in the active state. This is actually from the structure. So that means that if we were able to replace this tryptophan for something that is not fluorescent, okay, that is not quenched, sorry, for the Qvbr, then, for example, an alanine, we eliminate completely the signal, and what we get is essentially a linear signal, which is probably due to some electrochromic effect of the dye. and in fact it's in the other direction. So that gives you now the background to start changing, putting tryptophans in the path, okay? So here's an example. If we put the tryptophan in position 247 here, which is in the second transmembrane segment, we get a signal which is in the opposite direction. It's just unquenching here, unquenching depending on the voltage, okay? And that's the time constant of those signals. So we did that for many places, and I'm going to just give you the summary. Here is the summary of many of these residues. Some of them produce quenching, some of them produce unquenching, which actually tell us, and actually is color-coded. The unquenching one are the violet, the quenching one are green. So that means that now we can trace the movement of the Qvbr, in this case the first charge replaced by Qvbr, according to this arrow here. So there is actually a translation and a rotation of the first charge in the path. Looking from the top, you can see there is a very clear rotation of the first charge. It's important to notice that F290 here, it does not get affected at all. And it's not really F290, has been called the main hydrophobic charge, but it's actually not seen by the Qvbr for at least for R1. R2 has a different pattern, of course. It is still a rotation, but it's a different path compared to the first charge. And in looking from the top, it's mainly a translation and not too much of a rotation, okay? And this is expected because the charges are not going to be doing the same that the whole S4 segment as expected, right? So here's an interesting result. The F290, which I'm going to mention again. F290 has been, is the bottom part of the hydrophobic plug is a phenylalanine, right? When we replace by tryptophan here, we see absolutely no signal for R1 Qvbr. So R1, the first charge, when we put the fluorescent probe there, we saw no signals, right? However, if we hyperpolarize a lot, say minus 160 millivolts, we see a slow signal coming up. And so this R1 actually is moving much more deeper into the protein when you go very negative. And now this is important because if you remember some of our previous results, we were able to, in the closest state, to cross-link the 287 with 362. And that would mean that at that position, the charge is not really even getting close to F290, which is over here, as I indicated with the pointer. So what is shown here that we have multiple closest states. And when you go very negative, you go and populate more and more of this closest state, what has been called the Goldman Shift. Okay, so essentially what we have seen here is the physical basis of a Goldman Shift. In fact, if we look at the R1, the charge in the F290, as we move under extreme hyperpolarization, then it gets lower. The signal gets lower, as you expect, because you have to travel more of the transition between the closest state before you get to the active state, okay? Now, another important point here is the trap path that the charge moves in the active, coming back to the passive, the same. So we did that by changing the protocol. Here we look at the red traces compared to the violet traces, which correspond to the movement of the charge at the beginning of the pulse as compared to the end of the pulse, okay? And this is for 3 of these positions, 247, 320, and 326 for R1. And the fluorescence here is also showing the same thing, that in fact the path that follows as a function of voltage is not the same path for the activation as compared to the deactivation, all right? And this is also very nicely seen in the case of the time constant, which in fact the time constant in some extreme case like a 326W, you can see that the activation time constant is different from the deactivation time constant, That is for a short pulse. But now what happens if you just hold the membrane, which we call going into the relaxed state? The shifts are even larger. Okay, so the activation here is these violet traces, and the relaxed state are those black traces. A huge shift in the normalized Q and also in the fluorescence. All right. Good. So far this is the first and second charge movement. So first and second charges as they move in this different path. Now let's ask the next question, which is how this gating current couples to the ionic current. So in other words, the coupling of the voltage sensor domain to the pore domain. So here's a picture simplified of two subunits as we know today, that how this thing may happen. And it is very clear today that what happens is that when the S4 here moves, the S6 actually opens, and then when it opens, then ion conduction occurs. So this is called the canonical wave, which is an electromechanical coupling that is S4 is coupled to the S6 by way of the S3, S4, S5 linker, okay, which is this part here. Now, this is one way, but I'm going to show you that there is a different thing that came out lately that actually is pointing to a different way that the coupling may occur. This is a result that is at odds with the canonical path. So let me remind you about the mutation in the pore, which is a tryptophan replacing a phenylalanine in position 434, what we call the W434F mutation, which eliminates ion conduction, which is very useful to measure gating currents, right? Because what it does is that it makes the inactivation so fast that in fact the gating currents are visible and the ionic current get completely out of the way. Simple way to measure gating current. But now here we have a problem. You see, if we do the experiment in this particular mutant, which is L361R, we have a large shift to the left of the conductance. You see, the conductance turns on around minus 100 millivolts, which is not normal, okay? And then it just saturates. But then if you look at the gating current, the gating current actually now crosses the curve of the conductance, which is completely unusual. That is not correct. So this was the red light for us. So what we did was actually do a different experiment. Instead of using the W434 mutant, we actually depleted the old side from potassium and measured the gating current in zero potassium in both sides. And in this case, the gating current actually does show a shift, okay, to the left as it should be. So here it's comparing the blue trace is depleted, and this is the Qvbr of W434F. So now, if you just take the depleted case instead of W4, the Qv and the Gv show the normal situation. The Gv is shifted to the right with respect to the Qv. So here's an interesting situation. We have a mutation done in the pore, in the voltage sensor in the pore, that actually is affecting the voltage sensor backwards, right? So we think that this is due to the fact that you have an interface between the voltage sensor here, which is pictured in blue, with the red part here, which is the pore region. There's a lot of surface contact here. Notice that in these channels, like the shaker, the blue trace, the blue part here corresponds to the voltage sensor, and the pore here are actually translocated. In other words, there is a different interface between the voltage sensor and the pore depending on the unit. In other words, the subunit blue has contact with subunit red in the case of the shaker potassium channel. And in this here on the right, it's showing here the position 369, which is in contact with 412. 412 come to be a residue in the S5 segment, okay? And they're very close. In fact, we'll see later that they're all in van der Waals contacts, and that's really what we think is the reason of the interaction. So to understand this, what we did was to make tandem dimers. Here's a potassium channel with 4 different subunits here, okay, all different colors. But then we did a dimer in which we have 2 of those subunits attached together covalently. So then we inject the RNA for this protomer, this subunit, which are 2 subunits in fact, and form a channel. And the channel works the same. Here is a typical, inactivation curve and the recovery for inactivation. And here, of course, you have 2 possibilities because you can form it by putting the protomer 1 and protomer 2 in different orders, right? And what we found is that when you have 4 W434F, you have less inactivation than when you have 2 W434F, which is the case of what we are doing here, introducing 1 W434F in one of the protomers. So here's the point. What we want to do now is to see if we do some changes in the pore domain, in the way that pore domain is coupled to the voltage sensor, that actually makes a reflection into the movement of this coupling that is non-canonical. So what we did here is to use a very interesting mutation that was described by Rick Aldrich a long time ago. It is a triple mutation in the S4 segment that what it does, it makes the QV split in 2 parts, okay? And makes the GV, the conductance, shifted a lot to the right. And the second part of this conductance, the GV, actually goes with respect to the second part of the QV. These are about 100 millivolt shift. This is a huge shift. So what we did then was to make this ILT mutant and the W434 mutant in the called the near case or the far case by just changing the position in the protomer, okay? So the near case, you see the mutation, the ILT mutation, which I'm pointing with my pointer here, is close to the mutation W44S because they are in the alternate in consecutive subunits. Okay, in the far case, you see the W434F here, and here the ILT, so they are far away. Okay, so the question is, does that make a difference? And it does. So here you have the situation of again inactivation and recovery from and testing of inactivation for a long pulse. And let me show you the important point here. I always show 2 records because you have 2 different ways to put dimers, but in fact the results are very similar to each one. So I'm concentrating on the top one. First of all, let me show you the black traces here is the activation. The black trace here is the inactivation. Okay. Now, when you do the ILT mutation, of course, there is a huge shift, but the huge shift is only in the near case, which is here. You see the near case. And also inactivation develop a second component here. In the far case, which is the green, there is much less of a shift and there is no change in inactivation. So here we have a case that we have shown that the proximity is what matters here. So we went further here, and the QV also is actually important to show here, is that in the near case, you see the second component inactivation is actually related to the second part of the QV curve, right? Now, what we did next was to actually do many different mutations that affect the position of the GV and the QV curve. And when you do this in these dimers and you separate the far case from the near case, you can see right away here that in all cases where there is near, you have a huge span of the GVs and inactivations, okay, inactivation in the case and GVs, while in the far case, there's almost no change. In other words, all these mutations are affecting not through the normal pathway, but through the pathway between the S4 and the S5 segment, which propagate to the pore. Now, let's go on and try and understand what is this path. The first thing that we found that this position 412, which is in the S5, is very close to one of the residues that is in the open position in the voltage sensor. And in fact, when you put that serine into an alanine, you see a big shift of the conductance and the Qv. So the anomaly that we knew with the alpha 361 arc, which you remember was the crossing, if you replace this S4112 by an alanine, you get no crossing anymore. In other words, you have interrupted this pathway. So here comes an important point, is that the only one that actually makes this interaction. So we look at the structure, and what you can see here is that between the S4 on the left here and the S5 and S6 and the pore, there is a series of residues which are all in van der Waals contacts, okay? In principle, we actually took an account of 409, 411, 412, 433, and 434. You see, in the top, you can see these residues here, which are the S4, actually they are movable because the S4 moves. They're in contact with 412, 409, and those are in contact all the way to the selectivity filter. So here comes the thing. Can we test all those? So what we did was here, go back to our mutation that actually shows this anomaly, okay, which is the R361R. You can see the normal gating and conductance for shaker. And on the right, you can see the shaker now with this mutation where you see the anomaly. So this is just a repeat of what I said before. So now what we do is change each one of those residues that I taught you about by an alanine, which makes now the contacts much less because the van der Waals space is reduced. And in all cases, we see a recovery of the normal behavior between the activation and the conductance and the Qv curve. So that's very encouraging. We know now that any one of those that you replace, they work. So let me show you another non-canonical result. If you replace the leucine 366, which is part of the S4 by the histidine, the W434F, which remember is non-conducting, becomes conductive. And you can see here in 12 millimolar or 120 millimolar. It's not totally conductive, but it's much, you see, is you block with 4-aminopyridine, it disappears. You only get gating current. And you can see here the different cases with high potassium and then low potassium, and then when you have the block by the 4-aminopyridine. So in other words, this mutation in 366 makes the W434F channel, which is conductive, make it non-conductive, okay? So to repeat this, 366 with W434F produce conduction besides seeing the gating currents at the beginning. Now, if we replace 412, again, the first one in the series, by an alanine, this conduction disappears. So in other words, we restore the normal situation in the channel. So again, it's part of the chain, okay? And here you have a superposition of the S412A compared to S412, which is the wild type, okay? And here the ionic currents, which you don't need to go in detail on that. So now, we're going to disrupt the non-canonical coupling in the case of this L366H, one link at a time. So here's the case of the L366H 434F, where you have gating and ionic current at the same time, okay? Now, if you replace 409, the ionic current disappear, 411 to an alanine disappear, ionic current disappear in 412. So again, all those links of the chain, any one that you change, immediately stop this connection between the pore domain and the voltage sensor domain. So now comes the question, well, any link in the chain is important, but do they actually interact? So what we did was going into thermodynamic cycle for at least two of those. We actually concentrated in 411 and 433, which are not really very close, okay? And the way to do that is follow the technique that Baron Chan designed a while ago, which is actually to measure the energy of activation by measuring the gating currents of each one of these mutants. What you do is you measure the gating current, measure the charge, and then calculate the V median, which is the area of just to get to half of the charge, okay? And then the voltage for that is called the V median. With that, you can calculate exactly how much the energy of activation for that particular mutant. So you can calculate this energy for each one of these mutants, 433, 411, and then the complex, the two of them, and compare the energies. And here's the results. And you can see that, in fact, there is interaction because you don't add those two things. You get an extra energy of 1.94, I mean, essentially 2 kilocalories per mole extra when you put the two mutations. But one thing that we found here is that this experiment was not done with W434F. It was done with another way to block the pore, which is on the bottom part of the channel. In other words, not in the pore. It's on the band crossing. That's what we call 478 mutation. So what we learn here is the non-canonical chain affect the voltage sensor domain in the absence of W434F. That makes a very important step because now that means that the non-canonical coupling is also occurring in the normal channel. So in summary, what we have found is that the normal channel has two ways to couple the voltage sensing domain with the pore. One is through the S4 linker, and the other one is via a contact between the S4 and the S5 through a series of residues that ends in the pore by making this pore just conductive or non-conductive, which is the case of the slow inactivation in the shaker potassium channel. So let me conclude by saying that first, that the gating charge in the voltage sensor are translated across 10 residues of hydrophobic plug, which is specific residues influencing wells and peaks of the energy landscape of the voltage sensor. As I told you before, I would like to emphasize residue 287 in the case of the shaker, because that residue in the case of the sodium channel is not a leucine, but it's actually a threonine, making it faster. So actually that makes the difference between fast and slow. Next, I show you that the R1 and R2, which are the first two charges in the four charges that are important for the voltage sensor operation, have different trajectories. And those trajectories are different for the on or the activation as compared to the deactivation, indicating that it's not that simple that this thing moves back and forth like it was proposed before. It's a bit more complicated how the trajectory occurs. Then I show you that the voltage sensor domain controls the pore domain via two pathways. One, the second pathway, which is the one that I really have time to explain in more detail, is a non-canonical path that starts at the interface between S4 and S5 segment. The pathway comprises these four residues, 409, 411, 412, and 433, which span between S5 and S6 and the pore. And then I would say that residue 411 and 433 do interact. And finally, the important thing is that the chain of residue of the path influences the VSD operation. In other words, the translation is not from the path, from the voltage sensor to the pore domain. The pore domain talks back to the voltage sensor via this pathway. And with this, I would like to thank you, and I'm ready to receive any questions.
Okay, thanks, Francisco, for this very, very interesting talk. And it's time for a few questions. So the first is from Alberto Giacomello himself. So he was wondering whether this non-canonical opening mechanism is expected to be at work in different voltage-gated channel as well.
Yes, I think that it is. We think that we have described this very well in the case of the shaker, right? Because it's very easy to do this experiment. But we have some indication that the sodium channel has the same situation. And now these are channels which are called swap, in which one domain of the one voltage sensor domain is actually looking at the subunit of the other of the port domain of the other subunit. But there are some channels like HERC, for example, and others like BK2, which they don't have this swapping. And so it may be quite possible that in those, this mechanism is a very important mechanism for opening and closing and inactivating the channel. Yes.
Okay, thanks. And then Gonzalo Paulo, which asks you, do you have an idea of the mechanism that causes the hysteresis of the trajectories of the R1 and R2 chirps?
Yes. I think that the reason for that is that the actual it's not, well, you could call it hysteresis, is a good name for that, but it somehow when you go into the open state, and sorry, let's talk about voltage sensor, the active state. Okay, sorry, the active state. What's happening in that case is that charges move and get into another position, right? Mm-hmm. Now that position actually is not the final position. They start moving into different states which are lower and lower energy, right? In other words, it's not just two-state model. If we were a two-state model, that would be no problem. Everything would come back exactly the same. So in other words, you have several other states which are lower energy, okay? And as time progresses, you get more and more of those channels into these new states, and then you start shifting more these hysteresis. Okay, yeah. We have some idea which residues are important for that. We have some idea, for example, that the S3S4 linker, in the case of shaker, has a large influence in the way that the relaxed state occurs, right?
Thanks. So Lucie Delemont would like to ask you, besides the interaction with the protein, do you think that the lipids of the membranes play a role in this non-canonical coupling? And if this is something that you can probe experimentally also?
I think it's possible. We have no direct evidence for that. However, it is possible to actually approach this problem with some markers in the lipid. However, Eduardo Peroso just came out with not too long ago with a new structure. An structure actually for the first time for CAT1, which is an inward rectifier from the plants. And this channel is very interesting because it opens in hyperpolarization and actually is a non-swap. That channel does have a lipid in it, and that we have been studying that lipid, and that lipid is fundamental for the coupling between the voltage sensor domain and the pore. It's coming up. Yes.
And Luigi Catacuzzeno asks you, if you mutate the canonical path, can you still observe the effects of the non-canonical path mutations?
You mean if we mutate the canonical? Well, this is something has been not very easy to do because the canonical path, well, actually, in a way we have, because the ILT mutation does that, okay? But doing things in the S3S4 linker, normally it just kills the protein very much. So what we really would like to do is to actually interact completely the canonical path and see where you still can open through the non-canonical path. And I think that it will be possible to do with the HERC channel. Yeah.
We see.
Sorry that I cannot answer that. We don't have the experience.
I think that we have maybe a couple of minutes for a very quick questions, or if there are no other questions, we could move in. Thanks again, Francisco, for this impressive way in which experiments can dissect this non-canonical coupling at the limit of the single molecular events. So thank you very much for this. And I think that we can, thanks again, Francisco.
Thank you so much to you too. Thank you.